Welcome to the first annual Symposium on Mitochondrial Health, Health Span, and Aging. Stress, Restoration, and Healthy Longevity by Elissa Epel.
This is exciting to see this Mitochondrial Health, Health Span, and Aging first annual symposium. This has clearly struck so much interest. We have hundreds of people watching remotely. And I just want to say how honored I am to speak at this conference and to thank Sonia and Monica, Bob, Rob, and Sandeep for organizing this and really elevating the field. I'm kind of a newly converted mitochondriac. I have been studying the dark side of stress and disease for many, many years and have... You will see some cameo appearances from mitochondria in my work. Most of it is in collaboration with Martin Picard, who is a young scientist who has devoted his entire life to understanding stress and mitochondrial function and healing. Works with pretty much all the mitochondriacs here. I'm so happy to follow Bob's talk. That was a remarkable deep dive into what's happening in the cell. And I'll be talking a little bit more about what's happening at a kind of systemic organismal level in the mind and in the nervous system. So, salutogenesis, origins of health. I think of this in terms of allostatic states. I worked early on with the late Bruce McEwen and think a lot about allostasis, how we are constantly adapting to our changing environment. And so we have different states that push our physiology into extremes. We might live in the middle, but I will argue that living in the middle, our allostatic baseline, is one of chronic stress, that we are rarely at a true baseline of relaxation and restoration. Even when we are asleep, it is often compromised. So not everyone, but I would say that in general, on average, that's where society is. So I'm going to first talk about toxic stress and how that impacts aging and rapid aging. And then talk about salutogenic allostatic states, how we have control over our allostatic states, and thus eventually our allostatic load, how we can, through lifestyle, through building in natural interventions, we can reach states of hormetic stress, positive stress, as well as deep rest. So I said it, deep rest. I'd like you to just check in and see where you're at right now in terms of your own nervous system vigilance, holding tension. Can you relax just a little bit more? You're going to I promise you'll be safe. I might show you how bad stress is, but I would suggest that even when you walked into this room, you were bringing in baggage that kept your cells in away from growth, maintenance, and repair processes, and in more vigilant, anticipatory stress. So we can, I tend to think in terms of aging and geroscience. How can we slow biological aging? We're all hopeful and waiting for that multi-component supplement or supplements. And while we are waiting, we don't have a magic bullet or pill or pills for slowing aging and increasing health span that has been proven to work in humans yet. We do have very tried and true control over our allostatic systems. So lifestyle stress, I love that we are focusing on nutrition and exercise as known ways to improve mitochondrial health. And what I'm going to show you is that moving around to hormetic stress and deep restorative states should be on your map. They have been reviews and meta-analyses of these mind-body practices. There's no reason we should be leaving them out at this point. There's no other practice that can reduce inflammation signaling, such as NF-kappa B in gene expression reliably over studies, than a good dose of some type of mind-body practice like meditation. So I will have a slide on that. So what promotes enhanced health? Just asking that question really does shift us toward looking at physiology differently. We're not going to change and find some salutogenic factor in the blood that we've never discovered. I wish we would, but we can have a different framework so we can answer different questions. I love this model. My colleagues at NCCIH are really trying to put salutogenesis on the map. This is a recent paper by very smart program officers, really just mapping out some basic concepts here. So we all know the chronic stress disease story pretty well, that we can live a lifestyle and increase our risk a little bit every day with exposome factors, unhealthy nutrition, sedentary lifestyle. Old news. We also know that there are natural aspects that we come with through training or temperament, emotional well-being, our sense of purpose, feeling supported, feeling life satisfaction, feeling positive emotions. These are protective. We don't know well the biology of emotional well-being, but we are getting there. There are things that stand out, such as sense of purpose that is related to lower inflammation across the lifespan and longevity. And then in the middle there you see the practices. So what I'm going to talk about are these allostatic states, hormetic stress and deep rest. But we're not going to get there yet. I have to drag you through a little bit of chronic stress physiology, where I've spent over 15 years with collaborators here. When I came as a postdoc, I was lucky to work with Elizabeth Blackburn, who's still here, and Julien, and the three of us looked at this question of whether chronic stress could promote accelerated biological aging in terms of telomeres, telomerase, oxidative stress. And that finding was questioned because it seemed radical, and there was this black box. How possibly could chronic stress lead to these? And it certainly did in this first study that we published over 20 years ago. And that finding has been replicated first in depression by Owen Wolkowitz, and in almost every major mental disorder, we see this syndrome of accelerated, not just telomere shortening, but hallmarks of aging. So these are 4 hallmarks of aging that are pretty easy to measure. As a clinical scientist, we can get these in blood. Epigenetic markers, we look at clocks, the telomeres, the orange caps at the ends of chromosomes, the pro-inflammatory states or inflammaging, and of course our friend, the mitochondria, or I'm going to say the network, the community of mitochondria. This is a system, but the question that's come up much of today is there really one driver? Is there one of these hallmarks of aging that's at the center of the system, a core etiology? And so I try not to put my, bet on any one horse and really try to study as many markers as I can. But I have to admit, this was a very, very exciting day, and hanging out with Martin Picard a lot, I'll just say we're turning our clinical studies toward different ways of looking at mitochondria and manipulating them with these mind-body practices. It's really a compelling driver, and I'll show you that in some studies where we looked at this in terms of telomere shortening. So chronic stress and early traumatic stress have been associated with accelerated aging in all 4 of these markers, not just in single studies, but now in reviews and meta-analyses. So we've already heard today how expensive stress is. And so if we just start there, this is important to understand what is the cost of chronic stress energetically. And in this paper, we really focus on not how stress affects mitochondria, but how mitochondria drive the stress response. We cannot mount a healthy stress response if our mitochondria are impaired. The mitochondria synthesize the steroid hormones. And so people with mitochondrial disorders have dysregulated stress responses. And a healthy stress response and good adaptation to stress is at the core of healthy aging or slower aging. So let me go on about that. I don't know what kind of emotional reaction you have to this, but I'm like, plug it in. What happened? Can you restore? How quickly can you restore? So we can think of energy resistance as that hitting the wall when you don't have any energy left. It feels terrible. This is one of the most common symptoms of most chronic diseases, and as well as of chronic stress, and of course people with mitochondrial disorders. And so we can understand that in part through cytokines like GDF-15 that are very much elevated in these disorders and associated with fatigue. This is one of my favorite studies. Most of what I'm going to show you is in whole people. This is in whole cells, cultured fibroblasts across the lifespan. And Natalia Balba-Alves did her dissertation on this with Martin Picard and basically did a slow drip of cortisol and examined how these exposed cells compared to control cells. The model was of uncertainty stress, low-grade, low-dose, chronic stress. And these cells responded in the most dramatic way. So first of all, they developed kind of an allostatic load of the cell. So that you get initial mitogenesis, bursts of reactive oxygen species, and hypermetabolism within the cell. This is not whole body, but within the cell, hypermetabolism. And then, toward the end of the life of the cell, then they start showing the hallmarks of aging. Rapid telomere attrition, reaching the Hayflick limit much sooner than control cells, as well as their epigenetic clock showed rapid aging. So this is, in a sense, reverse translational. This is what we see cross-sectionally in humans. This is what we think happens over time with low-grade chronic stress over years and decades. In a study, a different caregiver cohort study, we collected very high-stress moms of children with autism and other chronic conditions. Or actually, this was all moms of children with autism. We found they were one of our highest stress caregiving groups. And then low-stress control moms. There's no such thing as low-stress moms, but relative. And we looked at their blood, a mitochondrial index of energy capacity, the amount of enzymatic activity relative to the amount of mitochondrial content. And in this study, we found that healthy premenopausal women under chronic stress had dramatically low mitochondrial activity, for some comparable to people with genetic mitochondrial disorders. But there's quite a range, and people who coped differently and had more positive emotions, particularly the end of a stressful day, looked like controls. So I'm focusing less on the psychology of resilience, but there's a big story, a hopeful story there. So we also know that mitochondrial and telomere length, they're close together, they're best friends, they're talking to each other and interrelated. And work by Rhonda Pinho and others have shown a clear path that when telomeres get too short and reach a critical length, they send out P53, which suppresses PCG1, which we've heard about a lot today. And this is leading to less mitochondrial biogenesis and impaired mitochondrial function, which then leads to ROS and impaired telomere function. So there's this positive feedback loop between the two, and it goes both ways as well. So in this, you can just see that chronic stress, and particularly cortisol, we know creates excessive reactive oxygen species that can damage telomeres, and this can lead to senescent cells. Okay, so we tested this in live humans. That was a model, that was from model organisms. We tested this in women that we followed over several years. And so we were able to measure mitochondria function sensitively. We were able to measure telomerase activity in normal human cells sensitively. Thank you, Julien and Liz Blackburn. Very hard to do, but these assays have both been, let's just say, highly regulated and optimized. So in this study, chronic stress was related to lower mitochondrial function. But also, chronic stress over time was not directly related to telomere shortening, but indirectly through mitochondrial capacity or the energetic capacity of the women at baseline. So this fits that model I just showed you. Mitochondrial capacity fuels or reduces the ROS, and the opposite is true. So poor mitochondrial function increases the ROS. This is related to, over time, suppressed telomerase and accelerated telomere attrition. So in terms of direct paths, really the action was through impacting telomerase, and no direct effects of chronic stress except through mitochondrial health and telomerase. This was from our student from Mexico, Mauricio Guillen. Now, I just want to quickly point out that we can think about mitochondrial function in the short run. In people with lived long lives and reproductive critical periods, we must think of life course, and that's where a lot of the action is during development, when function can be shaped for the life course, but also during menopause and during puberty. So in a study, a cohort of women we've been following since they were 10, they're now 50 and they have children, we have shown, this is called the National Growth and Health Study, we've shown that early traumatic stress is associated with impaired adult aging, accelerated aging, through accelerated timing of puberty. And we're now going to follow them. We got an NIH grant. It was like a miracle. I couldn't believe it happened. To continue to follow these women, they're half black, half white. We'll look at racial disparities. So we see here that stress has effects over the lifetime, and that early trauma has a long shadow of effects. So in summary, chronic stress hastens cell aging through energy impairment, but this is not deterministic. There's no such thing as chronic stress. There are chronically stressful situations, and we may be stuck in them. And there are interventions and ways to break up chronic stress. For example, I have compiled a book of stress reduction activities, all empirically supported by research that reduce perceived stress within 5 or 10 minutes, both hormetic strategies and relaxing strategies. And it seems like we all know that, breathing works and nature immersion, this and that, but the question is, why don't we do it? And so we've done studies where we have people do these practices every day just for 3 weeks to look at the impact on slowing aging and on salutogenesis. So we'll get to that. But this is just a summary of these mind states. And so we enter these every state every day. We range through a stress response to deep rest, either during sleep or particularly during a mind-body activity, or engagement in something deeply restorative. So yellow mind, I painted for you, gave away that this is a very subtle chronic stress state that we want to either move up into hormetic stress or move down into blue or green or blue mind states of relaxation. And of course, the psychological mindset of threat is a determinant of what state we're in, and feeling safe is a prerequisite for us to enter green or blue mind states. Recovery, a resilient response to stress or recovery from stress is physiologically something important that we can measure, and pretty much one of the only things that researchers agree on. When we think about resilience, there's a lot of different definitions, but a return to baseline or a quicker recovery or growth all are what we're going with for the definition of resilience. So you can see the green responses with the more resilient response. You need the stress peak. We don't want to see impaired stress responses, but we want to see a quick return to baseline. So in this paper, I just lay out how these different stress responses over a lifetime can either speed up or slow down the rate of aging. And so we can think of like the optimal zone where we want to have the training, the positive stressors every day in a manageable form for reducing rate of aging. Okay, so let's get to positive stress. What doesn't kill you might make you stronger. If you get the inflection point right, intermittent, must be manageable. Engaging in short-term stressors with a threat mindset you might be nullifying the positive effect because you're inducing extra cortisol. We have, in humans, not that many options, but we need to think of more and we need to test more. We have hot stress, hyperthermia, cold stress, hypoxia stress, and of course exercise, which we've heard a lot about. And if you think of others, let me know. We collect these, we try to study them. Okay, so we have this multiplex stress response, and Rob, really want to make sure I get to the restorative part, but we're getting there. We've gotten through the dark ages of chronic toxic stress, and now we're looking at short-term stressors and how they might slow aging or relieve suffering, relieve depression, stress, anxiety. So in my search for looking for hormetic stressors, happened to come upon the Iceman, someone who's known as the Iceman. Both were speaking at a health and longevity conference, and this is a Dutchman explorer who has come upon a protocol, very handy for researchers, it's all manualized, of hypoxic breathing and ice-cold exposure. And boy, is this popular. Who here has heard of Wim Hof method? Yes. So that's a lot of you. I guess that comes with being interested in longevity and mitochondria, possibly. So we tested this protocol and compared it to known protocols, exercise, meditation, slow breathing. And we set up a kind of an incubator where we stress people out, we look at their biological stress response, have them do the practice every day for 15 minutes, and then we get to look at their blood and biology at the end. And we do this with people who have high stress and depression. And that's because we have, number one, their allostatic baseline is, of course, elevated, so they're already going to be having accelerated aging, so we can see changes better. And I love the title of one of Owen's papers is When Blue Turns to Gray, and it's about the syndrome, the biological syndrome of metabolic dysfunction that comes with depression and most psychiatric disorders. So we advertised for this study by asking for anyone interested in building their stress resilience. Of course, a very popular topic. We had filtered through many, many volunteers to get a sample who was very high in depression, but not clinically depressed. And we tested high-intensity interval training, Wim Hof breathing, mindfulness meditation, and then our control group was slow breathing, which was not a wise control group because it turned out to be one of the best conditions. But we are, in our next set of studies, we will have a no treatment control. All interventions reduce depression within 3 weeks from, roughly around 15%, for some people 20%. This is not that surprising. We can reduce perceptions of stress within minutes. We can help people feel better with these lifestyle practices if they'll do it, and if they'll do it consistently. And so in one of these studies, when we can know every day electronically and also support and prompt people, we had phenomenal adherence. But what we found was that while all of these hormetic stressors or deep rest stressors promoted some relief from depression, it was through different mechanisms. So we're still looking through gene expression and autonomic profiles, and today I'll just show you two early findings. One is that the slow breathing increased parasympathetic activity, vagal tone, during the slow breathing, of course, but also during the night. And the more heart rate variability went up, the more the depression went down. So it is a part of the mechanism. And, after hearing Bob, you got to wonder about where they were in the cell danger response and if we moved them, because we're moving around this basal vagal tone. In the hormetic stress condition, with this Wim Hof breathing, the more that they showed a stress response, a sympathetic response during their practice, we tested how well their body responded at the end of 3 weeks. So the more they showed a sympathetic activity, pre-ejection period, the more relief they had from depression and anxiety. Opposite mechanism. So we're training them in these different mechanisms. When we look at the blood, we can see that the Wim Hof method created Yes, more hypoxic-induced factor, almost like a manipulation check. Yes, that's a stress pathway. Is that good for them? I don't know. We also got a decrease in inflammation, NF-kappa B, so we're pretty excited about that. Again, this was kind of the grail, is to try to reduce inflammation in this easy, short way. Okay, so let's move. We're continuing these studies, testing different conditions. Next, we're going to combine hormetic stress plus deep rest within one condition, because we need both. But let's go just into what the biology of deep rest is and why that might relate to mitochondria. What is happening to energy balance when we rest? We dramatically reduce our energy demand. And so we're changing the messages to our cells to change their gene expression activity. And in one, so for example, when we compare what chronic stress does to restoration, we see that chronic stress creates, of course, one of the highest energetic demands of anything we can do. We mobilize a huge stress response within seconds and minutes. We change most biomarkers that we can measure have a dramatic change due to the stress response. And of course, when it's chronic, that means that we're also getting into changed allostatic baselines. So this decreases growth, maintenance, and repair processes that we see in our gene expression measures. And we also ask the question of what about the psychological or subjective experience of stress and of rest and restoration. And so that is kind of the next frontier to really understand those. In terms of restoration, we're decreasing energetic resistance. What does that mean? We are reducing the amount of ATP that we're using. And so our body is able to then repair and conduct mitogenesis and basically increase the balance. We're in energy balance. We're not spending more energy than we're able to create and make. So energy resistance, when it's high, creates, for example, GDF-15 and other stress responses. So we're when we restore, we're reversing that. Okay, so what does that look like when we lie down? What does the biology of that look like? What does blue mind state really do? Is it potent enough, or is this, something we have ignored because it's not biologically active? Well, it turns out that we can go from yellow mind to blue mind within days. And when we think about the biological changes, we're shifting people into growth, maintenance, repair, optimizing cellular health, salutogenesis, moving people from, of course, the acute stress response, but also that yellow mind state of anticipatory stress that's so hard to get out of, where people are ruminating, expecting, even unconsciously. Their body's nervous system can be vigilant. Can more time in restoration promote stress resilience and more GMR activity? So this is kind of hypothesized what the energy allocation might look like with deep rest states, that we are increasing GMR and we're spending a lot of less energy turning on genes for survival response. So in terms of the biology, we wrote this review looking at decades of studies on meditation, Qigong, any mind-body activity. It's a huge literature. Mostly people are trying to compare and say which horse wins in this horse race. But what we derive, and what many people would conclude after reading this literature, is that there is a similar fundamental deep rest response regardless of which practice. And so the body is agnostic. You want to choose what you like and what you'll do. But there's, in terms of cellular energetic balance, they all do the trick. And so we have a practice where you need to feel safe to engage. The breathing tends to always change, so it's slowing and more rhythmic. And then the cellular repair response might look like this, where the social mitochondria are then fusing. There's less oxidative stress, the yellow stars. And the telomerase activity is able to cap telomeres and conduct more, say, telomere stability or telomere maintenance. In one of our studies, we measured blood before and after a week on a meditation retreat. Half the people were there just walking around enjoying themselves, and half were in practice and learning skills. And we find that over the week, that gene expression changes so dramatically toward GMR and away from stress responses. But the change was in both groups. So again, the body's agnostic. It really likes relaxation. Go to a resort, don't bring your computer, eat healthy Ayurvedic diet, and you will have the restorative response. But the group that got the mind training showed less depression when we followed up almost a year later. They maintained benefits mentally. So these are just a few reviews, and there are many, many more showing that mind-body practices promote a suppression of pro-inflammatory activity that we see at the level of gene expression, not at the level of protein. That's a very mixed literature when we look at cytokines directly. But for example, NF-kappa B signaling. Variable studies find upregulation of growth maintenance repair genes and the mitochondrial regulatory genes are such a big set, but those often rise to the top as well. Okay, so what have I shown you? Hormetic stress, these short-term stressors, as we heard from Mark Mattson, promote in recovery more of the anti-aging responses. It depends what system you're looking at, but in general, a conserved response across eukaryotes is much more along the lines of having cleaned up free radicals or oxidative stress with chaperones, corrected damaged proteins, increased autophagy, increased telomerase and telomere repair. So this is something that we don't want to confuse at all with chronic stress. It's pretty much the opposite effect. Deep rest can also create some of these changes. I like to think of it as hormetic stress creates rejuvenation, because through the stress response, at least in the animal studies, we often see like stem cell stimulation and repair of organs with caloric restriction. We can see pretty dramatic things. With deep rest, we see restoration. We see housekeeping. We see more, I'll say like persistent, consistent repair mechanisms. And we can see that with the telomerase. So in reviews of meditation and telomeres don't change very easily, but the telomerase is upregulated in most studies. And both of these are states under our control. So we can theoretically control our level of energy resistance, our level of energetic capacity to make ATP. And we're not talking about waiting. If we had time, I would lead you through a deep rest practice before you left today. So let's do it. Okay, let's close our eyes. We're going to do it. I've left lots of extra time because I knew you would be a very willing guinea pigs here. Okay, so get comfortable in your seat. That's not my bell. I've got my bell on. We're okay. And allow yourself, if you're comfortable, to close your eyes. And you can wiggle around and try to get comfortable. You can stand up and lean against the wall if you're feeling pain or discomfort. And if we had a bit more time, lying down is an extremely powerful position. It's receptive, it's vulnerable, it tells your body that you're safe and you can relax. So you can do that supported. Oh, good. We have at least some brave people who are actually lying down. Let your chair support you. Put your hand on your belly and one on your chest. And first, just notice, what is your breathing rate? As you inhale and exhale naturally. We're going to take a few deeper inhales and exhalations together. So breathing in through the nose, and then slowly letting it out through pursed lips. A long, slow exhale. And again, inhaling through the nose. And slowly exhaling through the mouth as if you're blowing a bubble. I want you to continue breathing this way. And notice a pause at the bottom of the exhalation. Just a slight pause, a stillness. Allowing your nervous system to relax just a few % more, noticing any muscle tension and letting it go. Breathing into it. When we slow our breathing, we reduce our metabolic demand. And when our exhalation is long and slow, we increase our vagal tone, our parasympathetic activity. And you might even imagine the network of social mitochondria delighting in some of the signals you're sending them, being more connected, asking, is this a time we can turn on restoration? Stay within the cell. Don't go traveling into the cytoplasm because we're safe right now. Okay, when you're ready, the challenge is to keep breathing slowly. Keep that soft stance you have in your body. And then turn your attention back to the cats and choose which one. Which cat are you? So that was literally a few minutes, and we changed our nervous system. We started to change some of the signaling in our cells. We see differences. Well, there's immediate changes within gene expression and post-translational activity within a few hours. So if you just went on a half-day retreat, you're going to see cellular effects. But we see telomere lengthening within 3 weeks. At least in one study, we see telomerase within 1 week going up in these. In a state, what we think is a new allostatic baseline. When we stress someone out, we see telomerase goes up acutely. It's a stress-responsive enzyme. It's defending us from oxidative stress. It's defending the genome. So we see that in people. We see that in mouse studies. And the question is, can we have an allostatic baseline where we're not suppressing our daily telomerase, but enhancing it? And that's what the mitochondria can do for telomerase. That's what we think based on the basic literature. So we're dealing with a new type of stress, which is not necessarily new to humans, but we're just flooded with existential stress. So we have personal stress, and we also have global stress that enters our body through news, through media, through caring. And so this is creating a mental health epidemic, particularly in those who are most vulnerable and receptive to overwhelming news. And that is people who have the fewest years of experience of living and have seen the fewest historic periods of stress come and go, and that's our youth and our young adults. And it's quite dramatic when you look at the mental health rates of depression, suicidality, climate doom. So as a professor, many of us have gotten together to try to do what we can for this epidemic in our own backyard, and that is to teach resilience skills to young adults, to our undergraduates. They come into class. We've measured this intervention. We know that they come in with about 30% having what looks like clinical rates of anxiety and a lot of gloom and doom. And we teach them contemplative skills. We teach them rituals that we know help with emotion regulation. We teach them breathing techniques. We teach them worldviews so that they don't catastrophize, so that they have a longer view of time. And we teach that from many wisdom traditions, not just Buddhism. Buddhism's really the most helpful, but we make it very secular. And so we've done this now with almost 500 students across every single UC, and we know that being together is really important in the healing. And now we're looking at biosensing. We're looking at their sleep and their nervous system and their brain responses to emotional stimuli. Yes, thank you.
Stay tuned for the second annual Symposium on Mitochondrial Health, Health Span, and Aging.